We're going to continue with chapter 11 today, and we're going to talk about that response. What happens? Okay, it receives the information, it goes into the cell, and something happens, or some steps that occur, whatever. And now something's got to happen because of that. Okay, so it could, in this case, regulation of transcription. Transcription, remember, is making a copy of the DNA. There's got to be something going on because of that signal. Okay, and so now the cell is going to have some sort of response and send it out. Okay, so something comes in, that's reception. Something happens, that's transduction. And then in the cell, I should say, something happens in the cell. There's some steps, and then they send out their response. Okay, now there can be nuclear or there can be cytoplasmic. So it can happen in the nucleus or it can happen in the cytoplasm. If it's involved in the organelles, it's not going to go in the nucleus. Okay, so ultimately the signal transduction is going to lead to something happening inside that cell. Some sort of activity is going to happen in the cell because of getting that signal. Okay, and again, it might occur in the nucleus. If it's transcription, it's going to go in the nucleus because that's making a copy of the DNA so that you can repair something or you need something, whatever. It might happen in the cytoplasm. If it doesn't need the DNA or anything like that, then it's not going to go in there. It's going to happen in the cytoplasm. Okay, so many signaling pathways. Okay, again, this is they get the information and then something's going to happen. Their pathway is going to be the synthesis of enzymes or other proteins. Okay, because remember, we have thousands of enzymes. Enzymes control all the reactions that occur in our body. Well, we got to make them. So, that might be the signal to, make those enzymes so in the digestive system so that we can break down our fats or our proteins or whatever. They're going to turn those genes on or off. Again, that's what's going to happen in the nucleus. So if what they get a signal and what they want to happen is some sort of protein made or some sort of gene to be turned on or off, that's going to happen in the nucleus. Okay. And then the final activation molecule or activated molecule is going to signal that pathway. Okay, so that's a transcription factor. That means, okay, I'm going to open up part of this DNA and I'm going to copy it. That's what the messenger RNA does. It copies it and then it sends it out to wherever it needs to go to make the protein. Okay, signaling pathways can affect the overall behavior of the cell. Okay, they can say, hey, you're getting too big, it's time to divide, or we need new cells. Okay, that's another thing that can happen, and that's going to happen, it's going to go in the nucleus and say, all right, chromosomes, you need to be doubling so that we can divide. We're going to talk more about cell division in the next chapter, but that's one of the signals is it can be telling the cell it's time to divide. You're getting too big because cells can only get so big. So you're getting to the max amount, it's time to divide. Wouldn't that be nice if we could do that if we were people? When we get too big, we just divide, get rid of part of us? Yeah, doesn't happen though, right? Okay, so the response, what's going to happen? All right, the response may be very simple. Turn something on or turn something off. So it might just quit something, okay? There's 4 aspects to the signal regulation to consider, okay? When you're talking about what's going to happen, okay, are we amplifying the signal? Okay, and that's when I was talking like the secondary ones. We have the response. If it's not big enough, do we need to amplify it? Okay. What is the response? Is it very specific? Is it kind of a general thing? So again, that's going to regulate the response itself. Does it need to be, is it only react on a very certain cell or does it react on a bunch of different cells? And then the efficiency of the response. Okay. We need it to happen. We need to have, like with the epinephrine, we need it to happen right now. Okay. So is it going to affect your heart rate and your blood pressure and everything, all those things that need to be done? So you might have to do some scaffolding. You might have to have some other proteins to help on. So when you're looking at your response, does it need to be amplified? Is it very specific? Is it very efficient? And sometimes you just need to stop because you've gotten away. You're the deer and you've gotten away from whatever's trying to eat you. Well, you can't stay at that, amped up. You don't want your heart rate to stay up there and your blood pressure. You want to calm down. So you're going to need to terminate that signal as well. So you can turn it on, you can turn it off. And then while it's on, do we need to amplify it? Do we need to be more specific? Do we need help making it more efficient? And then we need to shut it off as well. So you got all these different things that can with the response. Okay. An enzyme cascades, it amplifies it. Okay. So that happens. Enzymes can come in and help amplify the cell's response. If it's not big enough, they can come in and help. Okay. At each step, the number of activated products is much greater than. So you start little and you keep amplifying and making it bigger and bigger and bigger and bigger. And if you need to do that, you've got proteins that'll help. So it depends on, again, the response, and does it need to be greater? And if it needs to be greater, then you'll have enzymes that'll come in and help with that. Okay, which is really amazing that all that happens in a cell. You think about it. Okay, so we want to know if it, we're going to make it efficient. Okay, so if we have these scaffolding proteins, they make it more efficient. They will take away if there's too many products or there's too much other stuff going on. They'll help make it more specific so that you don't have a lot of wasted. They're usually large protons, doing chemistry, large relay proteins, and they're going to help. There's more attachment and they make it more specific. They make it so it's more efficient so you don't have a lot of wasted products. Okay, they can increase the signal, these scaffolding proteins. So again, if they increase the signal, they can make it more efficient by grouping proteins together. So they have the same pathway. So you're not wasting making another pathway each time. They share the same pathway. Okay. In some cases, they may also help activate some relay proteins, some ones that help, again, so that you don't have to follow the same, make new pathways. They can follow the same ones. So when you're talking about sending a signal, if you need it to be amplified, if you need it to be more efficient, you can have other proteins that step in and help. Okay, here's an example. Okay, so we're signaled. Here's the molecule that comes in and attaches, and this is the receptor. All right, something's got to happen. We got to do something. Okay, if we just go by this, it's not big enough. Okay, we need it to be a bigger response. So you have the scaffolding protein, it adds more proteins onto it, and then there might be more and more, and makes it a bigger and bigger response. Sometimes this is all you need. But if it's, something bigger than that, maybe we need to add more proteins, and that'll help make the reaction or the response bigger. Okay. And then we have to terminate our signal. In other words, like I said, with the epinephrine, you're releasing that and your heart rate's going crazy and your blood pressure is way up, and it helped. You got away, you ran faster than you've ever run before, but you can't stay in that state. Okay, you'd end up having a heart attack because your heart likes to, jump up once in a while, but it doesn't want to stay there. So you have to have something to shut it off too. Okay, they have a ligand concentration. Again, remember, ligand can be a signal molecule, but you'll have fewer receptors that will be bound. So when the concentration falls, when you don't need as much, then that's what it'll do. And then the receptors will actually unconnect and become inactive again. So they'll disconnect from where they were bound. And now that you don't have that signal anymore, it'll stop. Okay. Okay. If you have cell signaling to multiple pathways, you have more than one pathway, they have something called apotheosis. I can't say it very well, but anyway, what it does is when the cells are affected or damaged, when they've reached the end of their life, some cells will just divide and you'll get more and more. Some won't. Okay, they have a programmed cell death, and they're doing a lot of research on this because if they can figure out how to shut that off, people won't age anymore. No more wrinkles, because we only live so long. And they're trying to shut this off so people can live longer. Yeah, whatever. But anyway, they're trying to see, because cells are programmed to live a certain length and then they die. And that's part of this multiple pathway. Okay. So the best understood type of this is called, again, apotheosis. I can't say it very well. Anyway, what they do, this is when the components of the cell are chopped up, there's vesicles that do this, and then they digest them. Remember the lysosomes, those are the organelles inside the cell that help with that? They're going to digest that cell so that it no longer exists. Because some cells live, they'll just divide and divide and divide. And when you're young, you get a lot more of that. But as you get older, cells stop working and they die. And again, that's why people age. So there's scavenger cells that eat up those digested cell components. Recycle them so that you can use them again, but in a different cell. Okay, so what apotheosis is prevents the enzymes from leaking out of that dying cell. You don't want those enzymes to leak out, because if they do, they'll damage the cells next to them. Well, if those cells aren't ready to die, you don't want to kill them. Okay, so it prevents, it takes inside that cell and it kills off the inside of the cell. So basically you just have an empty shell left. All right, that's what you want. You don't want anything to leak out. You got to keep that stuff inside, otherwise it leaks out and then it kills cells next to it. Okay, and here's an example. This cell is done. This is a healthy cell. We don't want the stuff inside there to leak into that. Then it would kill it. Okay, so they do have, here's an example with worms. They have like a triggered signal that activate a cascade of suicide proteins, they're called, and for them to die. Okay, it happens a lot of times, like with a worm, or like, you know, when you pull a worm apart and they grow back. Well, the part that, you know, they can kill part of that off so it doesn't work anymore, but regenerate the other part. So when a death signal is received, it stops this protein. It's CEN9. You don't have to worry about that, but it's a protein that's inactivated, and then it causes them, once that protein, this protein is done, you know, once it's inactivated, then the cell dies. Okay, and this, they found it in these soil worms. Again, they, you know, when they do these research, they're gonna do research on, you know, who cares about soil worms? You know what I'm saying? But they have the same, they go through the same processes, so it's a lot easier to deal with them. Okay, this happens in our fingers. Okay, this is when we're trying to get hands. And what happens is these cells in between, there is a protein in there that says, okay, cells, you're done. It's time for you to shrivel up and die. Well, and it happens in these little pathways. And guess what happens? We get the spaces in between our digits and our fingers are formed. If this doesn't happen, then we have, you know, webbed hands, I guess. I don't know. The fingers aren't formed because you need these cells in between to die. And they sculpt your hands. Kind of weird, but kind of cool if you think about it. Okay. Oh, that's it. All right. So we're done with this chapter, the cell communication part. We'll move on to chapter 12, which is cell division. All right. So good luck with your assignments, and I'll speak to you soon.